Good morning, everyone. Is my audio clear?
Good morning, sir. Yes, sir. It's clear, sir.
Okay, thanks. I'll just adjust my earphones once. Please wait. I hope I'm audible now as well, right? Confirm, please. One second.
Yes, sir.
Yes, okay, thank you. So we were learning about restriction endonucleases, our RNM systems. We reviewed about the discovery and several properties of them, including the sticky ends and sticky ends or 3' or 5' overhangs, 3' overhangs, and blunt ends. We have seen several of them. There are 4 base cutters, some of them are 6 base cutters, and there are 8 base cutters. So we had a little bit of tentativeness or in conclusion about isosygomers and neosygomers, and I would want to verify a little bit on that. Isosygomers are any enzymes that recognize the same sequence. It is only about recognition of the same site, or having the sequence is in the site is same. The difference, neosygomers are a type of isosygomers. Only thing is that neosygomers cleave at a different point, or different cleavage site is different. As you can see, these two would also qualify as neosygomers. So do these two, SMA1 and XMA1. Here KPN1 and ACC651. These pairs qualify as neosygomers because they recognize the same sequence but cut at different sites. So those that are those that are isosygomers and not neosygomers would be recognizing and cutting at the same site. I hope that is clear. And we have to remember that neosygomers are a subset of isosygomers, where recognition is sites for 2 different enzymes is same, but they cut at different points. So the rest of the isosygomers recognize and cut at the same site. We also discussed about methylation dependency. Methylation. I have a way of doing that. We discussed about sensitive, methylation sensitive, methylation insensitive, and methylation dependent. Okay, so here methylation sensitive, majority of the restriction endonucleases are methylation sensitive. That means if their target sequence is methylated, then they cannot cleave that site. BAMH1, EcoR1, KPN1, majority of them are methylation sensitive. Say methylation insensitive, the example is here that MSP1 can recognize can cleave irrespective of whether the recognition site is methylated or unmethylated. That is one way. You can take it as an example for methylation insensitive restriction endonuclease. Methylation dependent is that restriction enzyme can cleave only if its target site is methylated. If it is not methylated, it cannot cleave. The example I gave is DPN1. All these things will also come into are important for our analysis, so you just try to remember these things. And we discussed about probability of sites. 4 base cutters will have higher probability, whereas 6 base cutters will have lesser, and 8 base cutters, for example, will be even further less. We also discussed about to understand that or to realize that the base composition of genomes is not as random as we think it is. Okay, if it were completely absolutely random, irrespective of the species, we should be getting similar size fragments from all the genomes, but that's not the case. This indicates that the genome is not absolutely um, random distribution of nitrogenous species. One more thing I would also want to remember is the table here indicates an average fragment size. Okay, don't take that this is what there is nothing absolute. They have calculated the average. Even then, still it does give us some insights into the nature of genomes itself. So we also did this thing, or where are we with this? Yeah, we finished this, I
We
think.
did this one, sir. We got it as 8 as the answer
That's
since
right.
it's given as a 4 base cutter.
That's right.
Uh,
Yes, yes. We got this as the answer. I don't know if we have done this. I don't think we have done this one. Is that right? Or did we do this?
we
No,
were
You've
doing
I
it,
asked
gave
it.
it.
sir,
I
actually.
told you people to do. Yes, yes. Anybody got the answer for it? Did you try it or you didn't?
Yes, sir.
Okay. Can you tell me what is the order? We'll start from here. How many sites does A have? First thing, A will have 2 sites because it's a linear DNA and you have to think about circular and linear. If you're cutting with A and it is circular and you're getting 3 fragments, then it must be having 3 different restriction sites. But if it is linear, you just need 2 sites to make it into 3 fragments like this, right? So if it is linear, it must be 2 sites, A and A. And for B, also it will give rise to 3 fragments. This is also 3 fragments, right? 1, 2, 3, 4, and 5. 5 sites you get. Uh, so how do we do that? What is your answer? What do I start with here?
So after 4 bases, we have a restriction site for B,
Uh,
4 kb.
here 4 kb.
We have a site for B.
First is B. Is
Yes,
that
sir.
right? Okay.
Yes, sir. And then after one other base, we have A,
1 kb.
1 kb. Sorry, sir.
Yes, yes, chill.
And after 5 bases from A, we have another site for B, 5 kb.
Yes. 5 kb,
Then 3 kb A.
3 kb A,
We are left with two more kb.
2 kb. Okay, that's good. Thank you. I hope, do we all agree on this or are there variations to what you got? If not, we will assume this is the correct one. But first thing, I want you to remind what makes a restriction map. I told you 2 things. One is the order of sites and the distance. Second one is distance between any 2 consecutive sites. Right? Here the order is given as B first, then A, B, and A. And then here we are giving distance from each of them. I would also, one thing when you're solving restriction mapping, I'm going to put a competition for all of you without any, let's see how you will go about it. So when I give you A and B, never take one side for A and B cutting at the same place. Many a times over the years, we come some students will come up with this. Is it possible that A and B cut at the same side? If that is true, this must also be true, right? In this place of A, there should have been B also should be able to cut. And the same case would have gone with the A as well. Okay, so that is not a possibility. When you put in restriction mapping problems, you have to ensure that there are separate sides. And I will also give you something like this. Say, for example, I have a DNA, and these are all not answers or not a particular way of thinking, because in different classes or different batches of students, we see different ways of thinking. It's about imagination and putting it. I will also share some of the videos that are on YouTube. You please watch them all. Different people solve it differently. I think I don't want to enforce one way because it is your own way. But if you are able to get to the answer, that is a valid way. And that is what I want you to ensure. Say, if I have taken a DNA molecule, linear DNA molecule, say 3, 4, 5, 2, 1. I gave you a DNA molecule like this, and I'm saying these are the restriction sites. I'll take new ones. C, D, D, C. Then if I cut this DNA only with C, what will be the data that what will be the sizes of fragments? I would get 1, only C. So I'll get 3.
Only C, then it's 1, sir.
Yeah. 1, 3, and 9 plus 11. Right? If I cut only with C. That's right. Yeah. If I cut only with D, then I would get 3, that is from here, then 5, this one, and then 7. So if I cut it with C and D, what would I get? I will get 1, 2, 3, 4, and 5. I'm just putting them in ascending order. So this is the restriction map, and this is what you will get. This is a good puzzle because if I give you this data, can you construct the restriction map? Okay, with restriction map, I would never say until the end of this course that this is done. It's about thinking, and we have to practice several of these and continuously should do. Say, for example, if I don't do restriction map for 6 months, and all of a sudden I start doing it, then I would require time. And sometimes it's difficult to even explain how I'm getting to the answer. That means you need to just keep on practicing. Okay, so we will, until the end of this course, we will keep practicing different restriction mapping problems. If you come across any good different variety of restriction mapping problems, you're please welcome to post it on the Google Classroom. Okay, that will be a good way for sharing with things with others, and then it's nice. As I said, there will be an assignment, and there will be a competition, and there will be a gift. We will discuss a small gift for those first 3 students, but the competition I will tell you in a while. Okay, that's about restriction mapping. So this is one of the easiest one. There will be complex problems when I give the assignment. Okay. So I hope you also, for biotech, people must have heard about star activity and other bioengineering may not have read all these things, so we need to go a little bit slower so that everybody gets to used to the rDNA. So in star activity is sometimes a
Sir,
restriction enzyme. Yes, please.
sir, if you don't mind, could you go to that previous that restriction mapping question you gave? Sir, uh, so from that answer, what we've got from that 15 kb cut to enzymes A and B. So
Which one?
if I'm
This
not
one?
mistaken, like if I'm not mistaken, the A fragment that I mean the whole DNA fragment cut with A is just given three fragments. So the actual possible that probable of what fragments is divided, it's 5, 8, and 2, isn't it, sir? So that we are getting, so that while dividing with B, we would get it as 4, 5, and 6, and then A plus B is 1, 2, 3, 4, and 5.
Do you mean to say there is a problem in this error? There is an error
No,
in
sir.
this?
I just want to get it clarified, sir. That's not about the problem, sir. I just want to get that clarified. I was
Okay, okay.
just
Now
getting
you
it
tell
clear.
me again once again.
Yes, sir. So actually, since you yesterday mentioned as when it's cut with A, it's given
Yes,
as 2, 5, and 8 as the size of those fragments.
yes,
But the possibilities could be 2, 5, 8, 2, 8, 5, 8, 2, 5
yes,
like that.
yes,
Then the
exactly.
another one is then the other one which we I think it's 5, 8, and 2. And based on that 5, 8, and 2, and then we cut with B, we're getting this 4, 5, and 6. So based on that, I think A plus B is given as 1, 2, 3, 4, and 5. And then so while cutting with B, we keep that 5 fragment, that part alone, and that B is cut there. So it's 4 and 1. And then it's like, I think maybe in that order it's coming. So that's what I want to get it clarified.
Yeah, I mean, yesterday I think I was describing about the ways to think about it, and I was telling keep it, try all possibilities. If I remember, that is what I must have told you. Yes.
Yes,
So
sir.
258, or it could be 285, or it could be 528 and the reverse of it. Okay. And if this is for just for A alone, for B also you would have one way of doing. Say for example, I will just try to, I'm not going to solve it as such. This is for A, all the possibilities. I'm now going to look at AB. Okay, A plus B. What does it have? 1 and 2. 1 and 2 could be, no, sorry. This is here, 2 is here for A, 5 and 8. So 8 could be made from 5 and 3. I'm looking at A plus B. And 5 could be made from uh, 1 plus
1 and
A.
4 and 2 and 3, sir.
And 2 is just like that. But here it is pretty straightforward in a way. Okay, this might not be as easy all the time. Now we need to make this arrangement properly. Which of these is going to agree with your data, right? So, and also remember, sometimes there can be more than one possibility of restriction mapping, sometimes, okay, but not always. So keep that also in mind. So how do we arrange this? If I now, you can put it either way. This is for A, this is B, and a B. Should we just put it this way? Yeah, A and A. If this is 5, say this is B, then if you have, but yeah, there is also one more issue that you need to take care of. Upon, you should get 258 with when you cut only with A. How would we do it? 1, 4. If I put a B here, sorry, I'll put here. This is 3. Okay, I will just write it anyway, and then we will get to it. So is this answer correct or wrong? It will probably match with condition A, it will match with condition B, but it will not match with condition, sorry, it will not match with condition B. So I'm telling only the ways to think for it. So now probably I have to reorganize these things until probably I get this answer. Okay. I don't know if that is what you asked, but well, when we
Yes, sir. That was like that's kind of that doubt I had, but a little bit of a clarification. But yes, sir. Now I got it, sir. Since with that answer we got, uh, it is matching A as well properly. It's giving coming as 2, 5, and 8, and then for B it's coming the same. So it's 4, 5, and 6. Like 4, 5, and 6 in the sense it's arranged differently. It's like 4, 6, 5 something, but it's matching.
but that's
And then
wrong.
the same thing for.
No, this answer is wrong.
No, sir. The one we got. I mean, one
Earlier, yeah,
we
this
just
one is
discussed.
right.
That
Yeah.
yes, sir. This one I was asking, sir. Not the one you said no.
Okay, okay, that's fine then. That's good. But take it as a puzzle. Every game takes a little bit of time, and this is a game too.
Yes, sir.
Yes, good.
Thank you, sir.
Chill. I was talking about star activity. Say, for example, BAM H1. The recognition site is GGATCC. So we will write it full. So it is supposed to cut in this site and this site like this. Star activity is if there is a sequence, something like GAA, TCC. Okay, GGATTC. Is this BAM H1 sequence? It is not BAM H1 sequence, but it is very close to BAM H1 sequence. Especially, this is only the difference between BAM H1 site and the site that I have drawn here.
Because for BAM H1 we would have GGATCC.
That's right. So yeah, in this position, that is the only difference. Sometimes if the buffer conditions were a little bit not good, or if the pH is not very well, or the ionic strength is there, some restriction endonucleases may also cleave these sites like this. And that is called as star activity. We'll just see, say for example, we'll take eco R1, GG, GAA, TTC, CTT, I mean, I should read always here. So the cut site is here. This is for eco R1. And if there are any sequences matching it, TTA, something like this, then too, it might cleave. So when we are really doing in the lab, one should have taken a lot of care so that there will not be any kind of problems like star activity. Why are we discussing this even is when I said in every lab, in experiment when we do, we try to have things in our control. We want to be certain of it. But because of any of these things, if there is a problem on star activity, we would have gone through and cloned the gene of interest, and then we don't know if we got it full or not. Only we will know it after we have sequenced it probably. So when we are mixing things, when we make the buffers for storage or buffers for the reactions, working buffers, one has to be very careful about elevated pH and ionic strengths and so on. If you go to NAB and other websites where there is a whole database of restriction endonucleases, they will give you details about specific details about each endonuclease. There are many restriction endonucleases that do not have star activity, but there are known star activity, but there are some which have star activity as well. Okay, keep that also in your mind. So here we are. We have learned. Yes, please.
Sir, could you? Yes, sir. So, sir, since in this restriction under extreme condition known as star activity, so like if all of those things occur, then that the enzyme would cleave that altered sequence also, or like I'm not able to get that one because like is it all the conditions if happens then it could happen, or is it either one of them? Like it's either elevated pH or low ionic strength. Like I'm not able to get this concept properly.
Okay, as it is something like this. Sometimes different enzymes react differently to conditions. So this is difficult to say, but it is. One could assume that both of them could be influencing one particular restriction endonuclease. But we have to go to, if we want to when we design an experiment and we want to see which restriction endonuclease we want to use, and say for example this is where you want to cut. Okay, first thing, our limitation or the choice of restriction endonuclease will depend on the site that is here, the sequence that is present here, right? The second consideration is once that is figured out, say you have E restriction endonuclease E, F, G, H, then you have to figure out which one you want to use. If I find that I want to use I have to use one of it. In that case, I will buy something that is cheap and something that does not have star activity. And there is also another consideration. Sometimes you would want to do double digestion, meaning that you want to say, for example, this one is restriction site for EcoR1, and here I have restriction site BAMH1. So first I should have cut with EcoR1, and then I should have purified the DNA from there, and then used BAMH1. That is because the buffer of EcoR1 is not necessarily useful, may not work with BAMH1. I'm just giving an example. So that buffer compatibility is what you should require. Say, for example, if EcoR1 and BAMH1 work in the same buffer conditions, I could take the DNA and add both EcoR1 and BAMH1 and do double digestion. Okay, so these are the constraints that we have. And in that case, if the buffer has too much ionic strength, or if the buffer has basic pH, then I should have chosen some of the available options which may not be having star activity in those conditions. And that is the only consideration we should have, and that is why we are discussing this here. But some enzymes may be responding to elevated pH, may be showing star activity in one or more of these conditions.
Sir,
Okay?
so this explains like even in another some of those extreme conditions which we mentioned, like the pH is higher and the ionic strength is low. So the enzyme could cut those sequences which is little bit altered, which you've given as an example. That is due to that those two problems or those two constraints, if you would say.
Yes.
Okay, sir. Now I got it, sir. Thank you, sir.
Okay, chill. Good.
So one other doubt.
Yes, please.
So is there any specific reason why low ionic strength and elevated pH is the only reason of the star activity? Like why basic pH generally prefers this, or why
Okay.
these particular
Uh,
criteria?
yeah, the reason I am not, I'm not very clear about the specific details, but I will give you what our thought should be there. It all has to do, it all has to do with the interface, or you know about proteins, right? How do they behave? Their structure is determined by, first thing is water and the charged ions that they may be there. Okay, so when you say ionic strength or low pH, you're talking about H plus, OH ions, or sodium ions, and so on. Okay, that means the protein structure is one thing. Being in a conformation is determined by water. I mean, hydrophobic ones will be inside, hydrophilic ones will be outside. And if the aqueous phase, the ions, charged ions, the ions that are present in the surrounding also influence the structure of the protein. And now we are talking about DNA interacting, the restriction endonuclease interacting with the DNA. And there will be some interactions between the DNA and the restriction endonucleases. Restriction endonucleases are sequence specific, and the nitrogenous bases are in the middle of the in the middle of the DNA, isn't it? They are in the interior. They are exposed, and they can. That means first thing is the restriction endonucleases are interacting with the phosphate backbone, but they're also interacting with the bases. That's why they are sequence specific. But if we have these alterations in these ions, the interactions between the restriction endonucleases and the bases could be altered. Okay, that is how we should think of. Does it make any sense?
Yes, sir.
Okay, but if we have to specifically answer, I have to go into EcoR1 and talk about the interactions and how they fit and all. But like that, we cannot do for all restriction endonucleases. But generally, this is the concept. All proteins have a structure, and all proteins, the structure of the protein is determined by water, hydrophilic, hydrophobic, and then we also have charged ions. That's why do proteins, some of them become denatured at different pH and so on. And in this case, they may not be denatured, but the activities will be altered. Here, the activity we are looking for is sequence specific binding of restriction endonucleases to DNA. It's not just binding. Binding is one thing, and then the second one is for cleavage or endonuclease activity. That also needs to have certain groups, the interactions, and then cleavage should happen. So these ions might be altering those things. That's why it could be star activity. Okay.
Yes, sir. Thanks a lot, sir.
Yes. So now we have learned about restriction endonucleases. We will keep on learning as we do some of the problems and other things. I don't believe in any learning that happens instantaneously. If that is, then it might be even memory based also, it doesn't happen like that. We keep recollecting whatever we are learning. We keep practicing problems over the course, and that is how we will probably remember it for a longer time. So if I started this, the restriction endonuclease is saying, if I want to make a recombinant DNA, I should be able to cut the DNA. Now, after cutting, I have to join it, right? If you have a DNA like this, and you have another DNA molecule like this, now I want to make a recombinant like this. For this, I have cut the DNA in both the cases, but now I have to join the DNA, and only then the recombinant DNA will form. So during this process of making recombinant DNA, we have several enzymes that are used to alter or modify the DNA. And so we have to know all the repository of all the enzymes that we have at our disposal, and then we should be able to make a smartest way. And what do you mean by smartest way? It all, of course, depends on the availability within our lab. But the simplest or smartest way probably is some of the constraints you might want to remember, not necessarily that they are absolute. Least number of possible steps. Least number of steps is one way I look at, because it will save time. It will save the yield of the DNA. It will save the resources that we may have, enzymes and other things. Least number of steps is one way of doing things. And one should also take care of about the constraints. What are all the constraints that the lab has? Say, for example, I might give you a question that these are the only things you have, and you have to achieve this. How will you do that? And there is one thing that we will not compromise is the outcomes. Say if I said I want this highly specifically, I said that the recombinant DNA that you're making should have this, but should not have these, right? I'm defining what the outcome should be there. That will be the most important thing. This is what your goal is, and if possible, then you have to look at your constraints, and then you have to make a protocol or a scheme with least number of steps. Okay, and when we are also doing these things, we don't leave everything to luck or so. We should try to have it be certain of what is happening in each steps. Okay, so what we will learn is I forgot to mention isocardomers. Isocardomers is those restriction endonucleases which may recognize different sequences but give the ends, the overhangs should be are identical. Say I will take GGA TDC. This is BAMH1 and SAU3A is GATC. It's a 4-base cutter. GATC cuts here and this one cuts here. Okay, let's make this and then we'll move on. So this is the DNA. If you cut this DNA with BAMH1, you will end up with G, C, CT, AG, and GATCC this way. This is BAMH1 overhangs. It would be the 5' overhang. Now look at the products of SAU3A. You will get GATC, right? You are only bothered about what the overhang is. Read the overhang from 5' to 3'. We will read GATC. The overhang here is also GATC, GATC, and GATC. So BAMH1 and SAU3A, because they are giving rise to the same overhangs, they are called as isocardomers. And why is it important? Now, if I have a DNA that is having BAMH1 site, GGA, TCC, GG, and say for example, this is a DNA, and I have another DNA somewhere, gene of interest I want to clone into this site, the BAMH1 site. And if you have SAU3A sites here, you can cut the gene of interest with SAU3A on by either side and be able to, because the compatible ends are compatible between SAU3A and BAMH1, you can use it to link it there. You can use it to ligate there. That's why isocardomers are also important. But these are not some things that I don't want you to memorize, but you should know that there are restriction endonucleases, although they may differ in their sequence specificity and cleavage site, but they may give rise to the same overhangs. Say, for example, all the ones that give rise to flush ends, or what you call this is blunt ends, they all can be called as isocardomers because they end up giving blunt ends, isn't it? So if I cut one DNA with a blunt end enzyme, I should be the corresponding vector should also be cut with the blunt end resulting restriction endonuclease, and that's how you can use it. So there are some more things that we need to discuss today, and ligase, alkaline phosphatase, linkers and adapters. We'll see if we can fill it or not. For all the enzymes, so you should know why and when it is it required. The molecular mode of action. These are some things you would have studied before, These all come from biology or the basic science. And I would want you to just go back to your textbook and try to find those things. One of the important things you should know is their substrates. You have to memorize it. What are the substrates for DNA polymerase? DNTP and a template primer junction, right? Then binding to that, it will keep on adding more DNTP to it. If there is no DNTP, or if there is no primer template junction, then DNA polymerase cannot do anything about it, right? And eventually, we have to know advantages and applications for all the enzymes. So what we will do for recombinant DNA to generate recombinant DNA is we will cut a DNA molecule into fragments, and we have another DNA molecule with similar or identical overhang. We can mix them up and we have a DNA. Is it the DNA here is held by only the hydrogen bonds between these? How many hydrogen bonds are there? There are 8 hydrogen bonds, right? So this gap or nick is still there. So unless that nick is sealed, we cannot be, because 8 hydrogen bonds holding 2 long DNA molecules is not very stable. They will fall apart easily. If I said if there is a DNA molecule like this, where you have about 100 or 200 hydrogen bonds, that could be little more stable. But just 8 hydrogen bonds is something like this you have, and they're very, very close by. They will open up and close. So we have to seal this neck, and only then it will be stable. Can you all, can anybody tell me what is the difference between denaturation and degradation? Denaturation.
Sir, denaturation is the structure of the protein is
With DNA.
DNA.
Yeah.
Yes, sir. Like denaturation, like it's like the whole structure is or its functions have been impaired due to its
What function of DNA?
like from converting its from its basic gene level to its protein level while undergoing that dogma due to that changes in terms of pH or altered temperature or some of the external factors. If it's changed, then it's subject to denaturation.
Okay. Degradation. Hello. Are you there, Ajay? Is it
Sir,
degradation?
degradation, I think it's more of its structure change due to other chemicals, something like that, maybe.
Okay.
I'm not able to get the differentiation properly, but yeah, but degradation, I think, laymanly speaking, maybe due to subject to other chemicals or something like that.
Okay.
I'm not sure.
Yeah, that's fine. Thanks. Say denaturation here is breaking the hydrogen bonds. In DNA, when you're talking about nucleic acids or even proteins also, you can think, if a DNA becomes into single-stranded DNA like this, then that is denaturation. Degradation is if the same DNA breaks its phosphoester bonds here. That is degradation. Say if in proteins you can talk of if peptide bonds are broken, then it is degradation. And if the conformational state is lost, you can call it as denaturation. While the all the peptide bonds are fine. So denaturation is reversible. You can just mix those DNA molecules in a solution, and they can form double-stranded DNA just like that. Whereas from degradation to here, that is not, it's irreversible. Once the phosphodiester bonds are broken, you need enzymes to fix it. Okay, so it's a good way of remembering as well. So if we take a DNA molecule um, cut with a restriction endonuclease, and say, for example, if it were generating 5' overhangs, and you allow it to mix it, you allow the different molecules to mix, then base pairing will occur as well as Stelling through hydrogen bonds between them. And then you have a recombinant DNA molecule, and the association is intermolecular, meaning that these two different DNA molecules have bound together through hydrogen bonding or base pairing. Whereas intramolecular, that means within the same molecule, the same DNA molecule could twist around like this and form hydrogen bonds. Okay, when we mix, that is what will happen. Hydrogen bonds formation is reversible in DNA, in water, and many things. They form and break and form and break. It depends on several conditions. So we need to, if we want higher stability of these molecules, we should have been able to fix these gaps. And that is where we have this enzyme. The process is called as ligation. And we use the enzyme called ligase that we are all familiar. We'll just have a quick way of looking at it. DNA ligase, it undergoes, first step is adenylation, which means a binding of AMP, AMP to the enzyme, right? That is addition of AMP to an enzyme like DNA ligase here is called adenylation. If you said adenylation, that means adding only the nitrogenous base. Adenylation meaning AMP. So the ligase would become conjugated with this AMP. You can talk of it is actually it comes as ATP, and pyrophosphate is released away, and AMP is bound to conjugated to enzyme, and then the enzyme recognizes the nick, the phosphate. This is the 5' phosphate of the DNA, and it will donate that AMP to the phosphate group, and that the 3' OH that is present here will make a nucleophilic attack on this phosphate and form a phosphodiester bond here, and that is how the gap is sealed and AMP is released out. Okay, so the energy actually is thought to come from ATP here in the form of AMP, and that AMP is donated to the 5' phosphate group, and that there by doing so, it is a good substrate for this 3' OH to make an attack on the phosphate group here and form a phosphodiester bond, and now the DNA is complete. Okay. So there are two different enzymes. The T4 DNA ligases are used in DNA. Predominantly, they use T4 DNA ligase. Firstly, because the cofactor required is ATP, and it's easy to do that. And there is one more, that T4 DNA ligase can ligate blunt ends also. Okay, so if you are using blunt end DNA and you want it to be ligated, there is a problem. There is no force acting to put these two DNA molecules together. If you had sticky ends, for example, then you had at least those hydrogen bonds between these two, and then the DNA ligase could act upon it, and the facilitation of the reaction is easier. But if it is blunt ends, there is nothing to keep these two DNA molecules together. Then it falls onto a DNA ligase to do that. So that is why T4 DNA ligase, which is derived from a phage, T4 phage, and that has the ability to ligate even blunt ends effectively. Whereas E. coli DNA ligase is not so efficient in ligating blunt ends, but it can do good job with sticky ends. Okay, so predominantly, say if I have in some cases, there is also a situation where I have, this is the case. Say if there is a DNA like this. Okay, I'm giving you 3 different DNA molecules, A, B, and C. I'll put something else here. So I want only the ligation of just A and B. And I don't want ligation, sorry, I don't want ligation of A and B, but I want ligation of B and C. Okay, that is the constraint I'm giving you. This is the situation. There in a solution has 3 DNA molecules, A, B, C. Their edges or the ends are something like this. A has both blunt ends, B has one blunt end, and B on one side it has a compatible end with sticky end, which is same as on the or complementary to the sticky end of C. And I want B and C ligated, but I don't want A and B ligated. Then which ligase, which DNA ligase would you prefer? If I add, yes, somebody is answering, please go ahead.
E. coli.
Yes, that's right. T4 DNA ligase, if I have, it can ligate the blunt ends and it can ligate the sticky ends also. So I would prefer E. coli DNA ligase in that scenario, because if E. coli DNA ligase cannot or is inefficient, very inefficient in ligating blunt ends, whereas it's good enough with ligating sticky ends. Okay. Yes. There are other ways of doing also. I will discuss in the next class. I don't want to overshoot into your time. Any questions so far? May I know what is happening with the assignment? Have you started?